All right. So moving on here. So there's other movement through the plasma membrane. Remember, the membrane is selectively permeable, right? So it only wants to allow specific substances and molecules to pass through it, right? This is really there to maintain homeostasis of the cell, right? So there's homeostasis of an organism, but there's also homeostasis of the cell, right? So that's really what it's trying to do. So there are inside of the cell, specifically in the cytoplasm, there's a high concentration of enzymes, proteins, which, enzymes are proteins, glycogen, potassium ions, right? They're all in high concentration in the cytoplasm, right? High. Outside the cell, there's sodium, calcium, and chlorine, right? These are in high concentration outside the cells, right? So it's trying to keep everything in at once in and everything out for very specific reasons, basically to keep the cell alive, right? One thing to note here too, it helps with the cell volume, right? The cell membrane, right? Things moving through the cell membrane does this, right? Because the cell wants to maintain its volume consistently, and the main way it controls its cell volume is water concentration in the cytoplasm. And how it controls water concentration or amounts of water is moving of ions, right? Basically making a concentrated or diluted solution inside the cell. So being selectively permeable to maintain a constant cell volume is important. Now, there are molecules that can freely diffuse or move through the phospholipid bilayer, such as oxygen and carbon dioxide, and steroids, right? That's why steroids are pretty strong medicines, because they can move into cells freely without having to be regulated here. So they can move through freely, which is good, right? The cells need to breathe, right? You don't want to spend too much energy trying to move oxygen and carbon dioxide out for, metabolism and cellular respiration. So that's good, right? There are other large molecules that need transport through. Specifically, they're non-lipids, right? So they're polar molecules is the big thing there. And then that's when we have those transport membrane proteins. So there are passive transport membrane proteins. These are ones that are not expending ATP here. And remember, we're talking about now, we're kind of rehashing this a little bit here, but I'm trying to show here is that movement through the cell membrane is really trying to move particles through, either to have diffusion or osmosis, right, of water moving through in and out to maintain the cell volume, diffusion of signaling molecules or so forth, right? So a lot of these things are passive because what's going on is you're going from a higher concentration, right, to low, going from the party room to your room. You have active transport that requires ATP. So you actually need something to move something, like energy to move. So it's always moving from a lower concentration to a higher concentration. The other unique thing that the membranes can use to transport is vesicles. So those are like the little bubbles I pointed out in part 1, very early slides. They actually form little like capsules. And so you can do endocytosis, which brings something in, or exocytosis, which packages something and takes it out. There's other vascular transport, but they're just basically phospholipid membrane bound sacs. So diffusion here, really the definition of it is the movement of solutes from area of high to low concentration in solution. So solutes are something you dissolved in something else, say sodium and water, right? So example here, right? Salt, sodium is dissolved into water, so the solute is dissolved into solution, which is our water here, right? This creates a random motion of all the atoms, right? And the ions in the solution. But when you're trying to move from, one solution to the next through a membrane, you're always going from high to low, right? Really, they can move both directions, right? Because they're not really regulated, but you're going to have most of your movement is going from high to low. It might be a little complicated to understand, but you can go both directions, because really what's trying to happen here is you're trying to have equal concentration of your solute on either side of your membrane, right? There might be a lab of this. I believe there is a lab for this, showing this in your lab, talking about osmosis and diffusion, and you'll get to see what this looks like firsthand. But you're always going from high to low. You're trying to balance out both sides of the membrane here with your solute, right? So the concentration gradient, this is just vocabulary here. It's the concentration difference between 2 points. For us in the cell, it's either the outside of the cell compared to the inside of the cell, or inside to outside, right? So they will always move from high to low. Now, osmosis is really the diffusion of water. So diffusion, when we say general diffusion, it means like a solute in a solvent. So like sodium in water is moving, right, from one place to the next. But osmosis is actually talking about the water molecules themselves are moving across a membrane, right? Because what if you can't move the solute or the ion molecule? Well, then you just move the water and you balance out the water concentration on either side of that membrane, right? So water will always move from an area of low concentration of itself to a high area concentration, right? So high to low, right? High amount of water to low amount of water, right? Because you're trying to balance that out. How water moves is aquaporin. So these are specific transmembrane proteins that are moving water from the inside to the outside. Okay, so this is another example of those transmembrane proteins. Osmotic pressure, this is the force required to move the water from one side to the next. So there is a force required to do this. It's not just random, but this is coming up because there is an important concept here in cell biology that we'll talk about, and it's related to medicine as well. So there are some terms we need to understand here of, okay, why do we care? So in osmosis, right, there are solutions that are osmotic, so they have the same concentration of like solute particles, so they're equal pressures on either side of this invisible membrane, right? So really diffusion osmosis is not moving anywhere because it's balanced. You're in feng shui, the force is balanced. But when you have a higher concentration of the solute, you are hyperosmotic, as in you have a greater osmotic pressure. It's basically water is forcing it one way out of the cell. When you have a lesser concentration of the solute, right, you're hypoosmotic, so you have less pressure forcing the water in. So what does that look like? Well, for us, when we look at it with cells here. So isotonic, the cells aren't really changing shape here, like in B. And the example we always give is like red blood cells, right? Because they're sitting in like a water solution all the time. Hypertonic solutions, what happens here is cells shrink or crenation occurs, because when you place the cell in solution, the water moves out because there is a higher concentration of the solute outside of the cell than inside the cell. So the water wants to go from like low to high, so it's going to move out. Hypotonic lysis occurs because what happens is there's less water inside the cell. So water is going to move inside the cell to balance out the two sides. It's going to swell or make the cell very large and then burst, right? Because it's trying to balance out the concentration of water on either side of that plasma membrane. So that's why we really care about osmosis and these concepts here, because in terms of like our blood and our cells in the human body, the body's trying to maintain the homeostasis of our electrolytes, our salts, inside the cell, as well as the cell volume outside and inside, right, and the water concentrations, because it does not want to die on either side, right? It doesn't want to do that. Die of dehydration or die of, too much water, right? So you really want to maintain a perfect isotonic solution as much as possible inside the cell. We will stop the video there so we don't get too long here, and then we'll go into facilitated diffusion here.